All right, real quick, we're going to go over active transport. So, with passive transport, that was specifically not using energy or not using ATP. With active transport, we have to use ATP. This is one of the reasons why you need to eat the foods, because the foods gets turned into the ATPs by your mitochondria. That's why they're the powerhouse of the cell. Actually, they're the oxidative phosphorylation center of the cell, but I'll ruin your life about that later. So this requires the input of energy. Proteins transport substances using energy because they're going against a concentration gradient. I like to think about this as like a big sock drawer. So you open your sock drawer, it's full, you got like 10 more pairs of socks. So you got to use energy to shove all those socks in. This is how your nerves work. So if you are really, really low on energy, your nerves and cognitive function starts to go down because you don't have enough energy to power neuron transport. That's just one of the reasons. So with this, we use electrogenic pumps to generate voltage across a membrane. One example of this is a sodium potassium pump, and we talk about nerves next week. This is going to be like the most important. So with this, we're going to actively pump sodium out and actively pump potassium in to create a voltage gradient. Because if 3 positives go out and 2 positives go in, as we see here, that's going to create a net difference in electrical charge on either side of our nerve cell wall. We can also do this with proton pumps. So by generating ATP, we can actively pump out hydrogen ions to create a net difference in electrical charge that then powers another pump on the other side. This is going to be really important when we talk about cell respiration and photosynthesis. We can also use these for co-transport. So this is where a membrane enables downhill diffusion to drive uphill transport. So what that means is if we look at the proton pump up at the top here, we're inputting energy to pump out hydrogen ions. And then we have a sucrose hydrogen co-transporter. So the hydrogen wants to go back inside, and for every hydrogen that goes in, a sucrose can go in. So they both need to be there in order for the pump to work. So we have to actively pump out hydrogen to fuel this pump that's downstream from it in that membrane. So differences between passive and active transport. Passive transport, little to no energy, high to low, high concentration gradient to a low concentration gradient. So we're going to be diffusing down. If we look at active transport that does require energy or ATP, we're going from a low concentration to a high concentration. So we're pumping against the concentration gradient. So the different types to review: passive transport uses just straight diffusion, which goes across that membrane, or it uses facilitated diffusion, where it's using a protein channel, but the protein channel is only to get bigger stuff through that can't fit through the membrane, still not using energy. Active transport has to use a protein channel and it has to input energy because typically we're going to be pumping things against their natural gradient that they want to go by. One way that cells do this that's very important is osmoregulation. So once again, we learned before, osmo refers to water, and this is to control solute and water balance. The contractile vacuole in some organisms acts as a big pump and it forces water out. So think about an animal that lives in water. It's going to get too much water. It's going to like explode with water. So they actively have to use ATP to pump out extra water all the time so they don't become hypotonic and burst. There are some different kinds of transport as well that we'll just touch on because there's really like no good place to put any of them. And one of those is bulk transport. So this is transport of molecules in or out of our cell. So endocytosis would be eating these molecules. Exocytosis would be egesting these molecules or getting them out of your body or of your cells, I should say. So with endocytosis, what's happening is macromolecules that we need are going to come into contact with our cell membrane. Our membrane, I can't do that, is going to fold in And then imagine my hands detaching from my arms, and it creates a vesicle and goes inside of the cell. The opposite happens with exocytosis. So, for example, if we have the endoplasmic reticulum making something, the Golgi apparatus is going to package it into a vesicle. That vesicle is going to float up to the cell membrane, become one with the cell membrane, and then whoosh, those tagged proteins and lipids that got made by the different endoplasmic reticulums then get sent outside to the cells or areas of your body where they are needed. So, with this as well, we can further classify endocytosis into two separate parts, phagocytosis and pinocytosis. So phage or phag means to eat. So for example, a bacteriophage is a virus that only attacks bacteria. It's a bacteria eater. Pinocytosis refers to liquids, or different fluids. So if the cell is quote unquote drinking, it's going to put that liquid into a vesicle and then take it inside. We also do this down below, receptor-mediated endocytosis. So this is how we actually get things inside of our cells. So we have these things called ligands that we're going to talk about next week. And when they bind to certain cell receptors, your cell takes them inside because that's like a messenger. Okay. So it's like a secret message. It's like, oh, secret message. It takes it inside and then it does something with that information, whether it is to make something or alert your cell that, hey, there's an intruder inside or anything like that. So that's just a quick overview of some different types of active transport and some other ways that things can get in or out of our cells.